Good morning everyone. My name is Jerwin Barbarona, and today's morning I'll be discussing about the gabberellins, the plant regulator. Our learning objectives is to explain what gabberellins are and how they function in plants, describe their effect on plants growth, flowering, seed germination, and fruit development. And last one is to identify the practical agricultural uses of gabberellins in major crops. So what is gabberellins? Gabberellin is a natural occurring plant hormones belonging to the deterpenoid carboxylic acid group. Over 126 gabberellins have been identified in plant, fungi, and bacteria, but only some are biologically active in regulating plants' growth and development. So the most important active gabberellins such as GE1 and GE4 acts as indigenous plant hormones that promotes stem elongation, seed germination, flowering, and fruit development. So many inactive gabberellins can also become active after being converted by the plant through the metabolic process. And so now let us proceed to the chemical natures of gabberellins. So gabberellins or GAS are acidic plant hormones derived from the terpenoids pathway. So they are synthesized from acetyl CoA through the melavonic acid pathway, so which is important to biochemical pathway responsible for producing many plant compounds. So through a series enzymatic reaction, acetyl CoA is converted into gabberellins procure and eventually into active gabberellins. So all gabberellins shares common four rings hydrocarbon structures known as the 8 gabberellin ring system. So these ring structures serve as the basic backbone of all gabberellins molecule. So despite of having the same core structures, different gabberellins vary in their chemical composition, resulting in different in biological activity. So based on the number of carbon atom, gabberellins are divided into two major categories: the C20 gabberellins and C19 gabberellins. The C20 gabberellins contain all 20 carbon atom and usually act as a precursor during gabberellins biosynthesis. On the other hand, C19 gabberellins have lost one carbon atom during metabolism, and generally the biological active forms such as GE1, GE3, and GE4. So the activity of gabberellins is also influenced by functional group modification. So this includes the number of position, hydroxyl group, or OH group, as well as the presence of methyl group, CH3, and carboxyl group. Small changes in this functioning group can significantly affect how strongly gabberellins interact with the receptor and regulator plant growth. For example, gabberellins such as GA and GE4 differs in the number of locations by hydroxyl group, which influence their biological effectiveness. The presence or absence of methyl and carboxyl group can also determine whether gabberellins is active, inactive, or serve as a precursor.
Mechanism of action have GA perception, receptor repressor binding, ubiquitin tagging, degradation, and gene expression. The GA perception is the hormone GA binds to its receptor like a cave fitting into a lock. And the receptor repressor binding, the GA receptor complex grab onto the DELIA repressor and the brake that stop growth. And next is ubiquitin tagging. The cell attach tiny garbage tags or ubiquitin onto the repressor to mark it for destruction. The fourth one is degradation. The tag repressor is shredded by the cell's recycling machine or proteasomes removing the brake. And the last one is gene expression. With the brake gone, growth promoting genes to turn on, causing stem growth, seed sprouting, and flowering. The major physiological function is seed germination, stem elongation and bolting, flowering and sex expression, fruit growth and parthenocarpy, and delaying senescence. The first one is seed germination. is a process where a seed begins to grow into a new plant. Plant hormones help break seed dormancy and allowing the embryo to absorb water, sprout, and develop roots and shoots under the favorable conditions. The second one is stem elongation and bolting. It is the process to increase in stem length, helping the plant grow taller and receive more sunlight. Bolting is the rapid growth of the flowering stem, especially in plants like lettuce and cabbage before flowers and seeds are produced. The third one is flowering and sex expression. The plant's hormones regulate the formation of flowers and influence whether male, female, or both types of flower develop. This is important for successful pollination and reproduction. The fourth one is fruit growth and parthenocarpy. The plant hormones promote the fruit growth after pollination, and they can also cause parthenocarpy, which is the development of fruits without fertilization, resulting in seedless fruits such as seedless tomatoes or cucumbers. The fifth one is delaying senescence. Senescence is the natural aging process of plants. Some plants' hormones delay this process by keeping leaves green, maintaining chlorophyll, and allowing the plant to continue photosynthesis for a longer period. This improves crop quality and extends their shelf. These physiological functions show how plant hormones control important stages of plant growth and development from seed germination, stem growth, and flowering to fruit production and delaying aging, helping plant grow healthier and produce better yields.
Hey everyone, today I will discuss the effects on crop growth and development. Plant growth regulators help improve the growth and productivity of crops. They can increase plant height, making plants stronger and healthier. They also improve seed germinations, which helps more seeds grow successfully. Another benefit is enhancing flowering, allowing plants to produce more flowers. This leads to larger fruits and better fruit quality. As a result, farmers can achieve a higher crop yield and better harvest. Plant growth regulators also help in synchronizing flowering and fruit maturity, which means crops can be harvested at the same time, making farm management easier and more efficient. To the recent research articles about gibberellic acid or GA, an important plant growth regulators, according to Singh et al. 2024, gibberellic acid act as a multifaceted modulators that supports plant growth, development, and stress mitigations. Their studies found that GA helps improves plant growth, enhances fruit qualities, increases stress tolerance, and supports sustainable crop productions. Another studies by Barton et al. 2025 highlights the role of gibberellin in extending the green revolutions. The research shows that GA helps regulate plant architectures, improves nutrient use, efficiencies, and can increase cereal crop yields. This finding shows that gibberellic acid is not only important for plant growth but also for helping crops adapt to environmental stresses and produce higher yields. This mix is a valuable tool for sustainable agriculture and food security.
Good day everyone, I'll be continuing our video presentation and my topic is advantages and limitation of gibberellin. And gibberellin are plant hormones that promote plant growth and development. They enhance fruit growth and can produce seedless fruit in some crops. They also accelerate seed germination, improve crop yield, increase stress tolerance, and delay senescence, which is the aging process of leaves and fruits. And gibberellin also have some limitation. Excessive use can weaken plant stem, causing them to latch or fall over. Their effectiveness depends on the correct dosage because too much or too little may reduce their benefits. They may also interfere with flowering in some plants, complicate tissue culture, and overuse may have negative environmental effects. And therefore, we should know the proper application is essential to maximize the benefits and minimizing the risk. Next slide is advantages and limitation of applying gibberellin. And these slides provide a more detailed look at the effects of gibberellins. Some major advantages include increasing fruit size, breaking seed dormancy to improve germination, promoting stem elongation, delaying fruit ripening, stimulating enzyme production during melting, inducing parthenocarpy or seedless fruit formation, and regulating flowering. On the other hand, there are also disadvantages. High concentration can cause lodging because the stem becomes too long or weak. Over-application may reduce fruit quality and increase production costs. Gibberellin can also suppress root growth, and their effects may vary depending on the crop species and environmental condition. And gibberellin are very effective, but they must be applied carefully and according to the recommended dosage. And next slide is agricultural application in different crops. This slide shows how gibberellin are used in different crops. In fruit crops such as grapes, they increase berry size. In citrus fruits, they delay the rain aging and improve appearance. In apple and pears, they promote parthenocarpy or resulting in seedless fruits. In cherries and peaches, they increase fruit size. For cereal crops, gibberellin stimulates seedless growth in rice and promotes alpha-amylase enzymes production in barley during melting process. In vegetable crops, gibberellin break potato tuber dormancy, encourage stem and leaf growth in celery and spinach, and influence flower development in cucumbers and squash. These applications show how gibberellins help improve crop growth, quality, and productivity. And next slide is a recommendation for gibberellin application. Recommendation for the proper use of gibberellin for farmers. It is important to use only the recommended concentration. Apply gibberellin at the correct growth stage. Combine them with good crops management practices and avoid excessive spraying to prevent waste and lodging. For researchers, future studies should focus on understanding crop specific responses, developing environmentally friendly formulations, studying how gibberellin interact with other plants' hormones, and exploring their role in helping crops adapt to climate change. By following this recommendation, gibberellin can be used safely and effective to improve agricultural production. And that's all my parts. Thank you.
So, in conclusion, gibberellins are among the most important plant growth regulators because they control seed germination, stem elongation, flowering, and fruit development. So, their agricultural application have significantly improved crop productivity and quality. However, proper application is essential to maximize the benefits while minimizing the unwanted effects. So, continued research will further expand the role in sustainable agriculture.